السلام عليكم. لا الصوت كذا واضح. Good morning everyone. I would like first to thank the organizing and scientific committee for giving me this opportunity to be part of this great scientific event. So today we will have a small presentation about some basic concepts in mucosal immunity. The outlines will be an introduction to mucosal immunity, and then we'll talk about mucosal associated lymphoid tissue, and we will have a couple of slides on secretory immunoglobulin IgA, and we will have basic concepts of oral tolerance, and then we will have some slides on some clinical implications about mucosal. So the main learning goal of this lecture that you will be able to describe the mucosal immune system. Okay, so we will talk about what is the mucosal surface. Mucosal surface can be generally divided into type 1 and type 2 mucosal surface. Part of the type 1 is those in the gut and the lungs, and type 2 are those usually found in vagina and eyes and mouth. Okay, when we talk about the mucosal surface itself as histology, it consists of a single layer of epithelial cell, a layer of connective tissue, which we call the lamina propria, and a thin muscle layer. The outside of the mucosa is usually lined with mucus. These mucus are usually made of high molecular mass glycoproteins. When we talk about the mucosal immunity, basic concept of the mucosal immunity, we can consider it as a thin skin. As skin is a quite thick and tough and implementable, mucosal surfaces are the major route of entry of pathogens in the body. Mucosal surfaces are continuously exposed to external infectious agents, as everyone knows. So specific immunological defenses at mucosal surfaces is mediated by a specialized arm of the immune system that is termed as the mucosal immune system. Some facts of the mucosal immunity is that the mucosal surface of the human body presents an area almost 100 times greater than that of the skin. So it's almost composed of almost 400 square meters of mucosal surface. It has been estimated that up to 80% of all immunoglobulin producing cells are physically located in the mucosal immune system, which give us that importance. The mucosa have a very complex immune system, functionally and anatomically different from the systemic immune system. It is capable of fighting pathogen, antigenic, and exhibiting tolerance to non-pathogenic antigens. And one of the special categories or special criteria of the mucosal system that not all areas are open to bacteria. So, for example, the lower airways are normally sterile compared to the upper respiratory tract or the gut. Okay, when we talk about, we will talk about some non-specific factors relevant to the mucosal immunity. One of them is the outward flow of secretions leading to diluting and flushing of non-wanted pathogens. Peristaltic ciliary movement leading to flushing and removal of unwanted agents. The acidic pH and bile salts found in the mucosal system lead to inhibition of growth of pathogens. The antibacterial molecules such as lysosomes, lactoperoxidase, defense The residents of commensals inhibit growth of pathogens, so this is one of the very important factors relevant to mucosal immunity. And also the mucosa itself, it helps to trap external particulates. When we talk about the mucosal immune system, we need to talk about the mucosal associated lymphoid tissue, which we usually call as MALT. MALT is a highly specialized immune system which protects mucosal surfaces, and it's considered to be the largest mammalian lymphoid organ system. And it has been estimated that in an adult, it's almost composed of 80% of all lymphocytes found in the human body. Much larger lymphoid cell content than the classical lymphoid system, and it has a very important dual function. So first, it has a defense against pathogens and prevention of response to commensal flora, food antigens, or inhaled antigens. So MALT has different sites in the body. The first one is called the nasal associated lymphoid tissue, which we abbreviate as NALT. And one of the most studied examples are the tonsils and adenoids. We have also the gut associated lymphoid tissue, which represented by the pair patches. We have also the BALT, broncho-associated lymphoid tissue, which can be found in the respiratory tract. And also we have the mammary and salivary glands and urogenital tract. Okay, so we will have a couple of slides on the major features of the mucosal immunity. The administration of antigens at one mucosal site results in specific antibody production to a distant mucosal site. And this can be very clear in examples of mucosal or mucosal vaccinations. Some regional preference seems to occur. For example, induction by NALT lead to more robust responses in respiratory sites than in gastrointestinal sites, and this has been clearly shown in different types of vaccines. The B cells in the mucosa are selectively induced to produce dimeric IgA rather than other isotypes such as IgG and IgM. The mucosal immune system has mainly three functions. The first one, it serves as the body's first line of defense from antigens and infection by preventing colonization and invasion by pathogens. It also preventing the system immune response to commensals, bacteria, and food antigens. This is what we mean by oral tolerance. And it has the ability to regulating appropriate immune responses to pathogens encountered on a daily basis. The main challenge that the mucosal immune system needs to deal with, that while the systemic immunity normally functions in a sterile environment and responds strongly to any foreign invaders, the MALT must be carefully select the nature of the response in order to avoid tissue damage and immunological exhaustion. So this is a very big challenge of the MALT. Okay, we will talk about the gut, which consists as the largest single lymphoid tissue system. It's anatomically composed of a single layer of epithelium joined by a tight junction allowing the passage of water and ions. It has brush border microvilli and dense layer of glycoperoxidase, additional protection. It has a thick mucus coat with secretory IgA traps and expels invading microorganisms. The effector component consists of intraepithelial lymphocytes and scattered lamina propria lymphocytes. And these mucosal surfaces, particularly in the intestines, are crucial sites of innate and adaptive immune regulations. one of the main majors of the mucosal immunity is the secretory IgA. It consists to be the predominant immunoglobulin in mucosal. This type of IgA. In the serum, for example, around 12% of IgA class, and they are primarily monomeric. However, in the mucosal, it's considered to be around 90%, and it's in the dimeric rather than monomeric IgA. As we mentioned, that the mucosa, these IgA are called secretory IgA, produced mainly by the malt plasma cells in the lamina propria. And these dimer IgA are bonded by a G chain, unlike serum IgA, which they are monomeric. Binds to a glycoprotein form of epithelial cells and does not activate complement pathways, so actions are non-inflammatory, and this is considered to be an ideal for protection of mucosal surface. Okay, so we'll be having a couple of points regarding the mechanisms of protections by these IgA at mucosal surfaces. So they have the ability of inhibiting any adherence of any pathogens. They have the ability to neutralize of enzymes, toxins, and viruses. Immune occlusion and inhibition of antigen absorption, and also they can bind to microbes and toxins in the lumen, preventing any attachment to epithelial cells and trapping them in mucus layer for extraction in stool. Also, these antigen bound to secretory IgA may be presented to the dendritic cells, which in turn present antigens to lymphocyte to initiate any immune response. As any other mucosals, any other immune site, there should be both ways, the innate and the adaptive innate responses. So when we talk about the innate defenses in the mucosal immunity, usually the innate immune cells, they have reduced levels of pattern recognition receptors, which we call them PRRs, at the epithelium level. There's no need for inflammatory T helper response for these ones, so it's prevented. And there is a very active induction of T regulatory cells that produce IL-10 and TGF-beta. And also one of the most important is the mechanisms of oral tolerance to not react to these commensals. On the other hand, why do these pathogens induce an immune response? They have the ability to adhere to epithelium and produce toxins. They use the M cells or dendritic cells to cross the epithelium, and there's an ability to activate, or they have the ability to inactivate phagocytosis to induce their apoptosis or prevent their recruitment. And if they invade the crypt, higher levels of PRRs are there to induce strong immune responses. Okay, so one of the more important concepts of the mucosal immunity is the oral tolerance. These oral tolerance are specific suppressions of the cellular or humoral immune responses to an antigen by prior oral administration. Unresponsiveness of T cells to ingested food proteins may be the result of 3 different mechanisms. It can be due to T cell energy, T cell deletion, or induction of Tregs at the oral entry site. there should be, or there are many factors influencing the induction of oral tolerance. Age of the host is very important. Nature of the antigen. Is it soluble or particulate? Is it a protein or a carbohydrate? The dose of the antigen administered, the nature of the antigen presenting cell, and other genetic factors related to the host itself. Okay, so some clinical implications about regarding mucosal immunity. IgA deficiency, for example, is the most common primary immune deficiency. It's usually diagnosed by a serum IgA concentration test. It increased the incidence of infection, upper and lower respiratory tract, GI tract also, higher incidence of autoimmune diseases, and higher incidence of allergic disease, and also in celiac disease. We also have the IBD, the inflammatory bowel disease, one of the major mucosal system disorders. It's, as you all know, it's a chronic inflammatory condition. It has two overlapping phenotypes. We have the Crohn's disease and ulcerative colitis. Why does it happen? Usually due to dysregulated mucosal immune responses to unknown antigen present into the normal bacterial flora. Some features of this IBD are they could be cell-mediated immunity, especially in active Crohn's disease. One of the main features is that they have high number of activated mucosal T cells secreting interferon gamma, which are usually T helper 1. We also have high mucosal production of cytokines that activate T helper 1 cells also by producing IL-12 and IL-18. And it has been documented that there are major defects in Tregs. On the other hand, the humoral immunity, we have high number of plasma cells producing IgG production mainly, and imbalanced in the pro and anti-inflammatory cytokines. One of the major diseases that has been marked in the mucosal is the celiac disease. The major features of it is the virus atrophy with lymphatic infiltrate, increased epithelial proliferation with crypt hyperplasia, and malabsorption. Looking at the celiac from immunological features. Yes.
Yeah, just 1 minute.
Okay, yeah, this is the last slide. The major immunological features are the antigen, which is known, the gluten. It has a strong association with HLA, DQ2 and DQ8, increased B cell activity, and it has been marked that celiac disease have high levels of antigliadine, anti-endometrial, and tissue transglutaminase. Just to summarize what we talked about, mucosal cells are continuously exposed to external infectious agents. Malt is the largest lymphoid organ. It has a dual function: defense against pathogens and prevention of response to commensal flora and food antigens. We had a couple of slides on malt sites. We talked about the NALT, GALT, and BALT. We said that the IgA is the predominant immunoglobulin found in mucosal surfaces, and we had a couple of points that why commensals doesn't induce an immune responses and why do pathogens induce an immune response. We had some points on the oral tolerance and a couple of slides on clinical implications. Thank you very much.
Thank you very much, Dr. Abdullah, for your great lecture. And only we are sorry for the delay that happened at the beginning of the lecture.
It's fine.
Now we have just 1 minute for questions.
Thank you, Dr. Adam.
Thank you very
What
much.
is the T cell allergy? I didn't understand this. This is, I think this is one mechanism of oral tolerance.
Yes.
What do you mean?
Okay, so T cell energy usually, usually T cells should respond directly to a foreign pathogen or an unwanted antigen. In the oral tolerance, there are mechanisms that enable the immune system to downregulate the T cell response through many mechanisms. The Treg is one of them. So basically, that it gives an order to the T cell to don't respond to this type of antigen. So it will not recognize it as a foreign, such as if the food we eat or the normal flora.
Thank you.
Okay.
Thank you for the lecture. Regarding celiac disease, what is the explanation of complication of lymphoma? Is there any underlying sequences?
I'm sorry, I don't have the answer for it. I'm not sure really. Okay.
Any more questions?
Okay, thank you.
Thank you.
We'll move to the next lecture.